So isoelectric focusing is a technique in which we try to separate a protein mixture on the basis of its charges that come as a result of its side chains on the protein surface. So the isoelectric point, which is Pi, defined as the pH at which the net charge is zero. As we can see in this chart over here, when the pH of the protein is below the Pi, then we will have a net positive charge. And in this scenario, we will receive this protein towards the very end of the gel when we run it upon the electric field. And if the pH is greater than the Pi, then there'll be an overall negative charge, and that would imply that your protein will stay towards the bottom of this figure. So as you can see in this figure, upon the application of an electric field, the molecules will go from the high pH to low pH depending on whatever their value is, the value of the Pi. This technique vastly differs from SDS page, where SDS page actually denatures the protein. So isoelectric point was not denaturing the protein. And over here, SDS page provides a detergent, which is sodium dodecyl sulfate, which disrupts the non-covalent bonds in the proteins and causes the molecule to unfold and become a rod shaped structure. Um, so these anions, that are the negatively charged unfolded proteins, will bind to the main peptide chain of SDS. So for every one SDS anion, two amino acid residues will be bound to it. And we can run this on a polyacrylamide gel and see the distribution depending on the overall charge of this protein. If we mix both of these techniques together, it is called 2D page, where we have a two-dimensional polyacrylamide gel electrophoresis, and the mixture of proteins is first run through isoelectric focusing, which separates the proteins based on the charge, and then the polyacrylamide gel electrophoresis allows this entire gel to be spread out on the basis of their molecular weight. We can create better resolution images of this broad pH gradient that we get into a more narrow range. For example, if the 2D page was run at a pH of 3 to 12, we can now run it for 6 to 8, which is like stretching the portion of this polyacrylamide gel where we want to focus. And we can stain and we can analyze it under different computational techniques to see the peak of the protein curves. A few important properties of this technique is that it is a standard. That means you can reproduce this particular protein spotting pattern in multiple experiments. Also, one thing we have to keep in mind is that the sensitivity of the protein staining depends on the method that we used, and also the resolution analysis computational technique that we use later on. And the proteins that are the least in pH or the most in pH will be very difficult to separate on the basis of molecular weight later on, and that is why they might be difficult to analyze. Another interesting protein separation technique is differential gel electrophoresis, which is DIGE, and it uses the labeling of proteins using different fluorophores. Fluorophores are special polyaromatic hydrocarbons that can absorb light to get excited and then emit another wavelength to return to ground state. So we label, say, a protein mixture with a CY dye. So Psi dye is basically a fluorophore, and we can provide it a label, and this label allows it to be seen later on when we run the gel electrophoresis. What we are basically trying to do is to migrate these proteins through a gel on the basis of their molecular weight and Pi, which is basically 2D page, and we will then be analyzing them together by labeling them with different fluorophores, and then analyzing the image of it by overlaying all these screens together. This technique is size and charge matched, which means that the same label protein from different samples will migrate to the same position regardless of the dye used in the different positions in the wells of the page. The polyacrylamide gel. This technique is also pH insensitive because the molecule, the fluorophore that we are using to label these molecules, it will not affect the charge of that molecule in a different way. So there's no change in signal over the wide pH range used during the first dimension separation, which is the isoelectric focusing, and then the consequent SDS gel. Moreover, we will be able to distinctly resolve all the signals of the fluorophores with high accuracy. We will also have a high sensitivity to the protein, and there will be a minimal loss of the signal during labeling, separation, and scanning because we're using one different gel, so there is no more bias or like on the basis of staining and other such manual operations. Just for a comparison, the advantages of 2D page and DIGE is that both of them use protein separation over a mass range. They also analyze large protein quantities, and the proteins can be excised or subjected to in-gel proteolysis and analyzed by mass spectrometry. And a disadvantage that both of these techniques also share is that they are not a good technique for the proteins available at the edges, which is the molecule with high molecular weight or high or low PI. And they are very expensive compared to the normal silver or Kamasi staining gels. Um, and also they are very time consuming because they might require 3 to 4 days per run, and they require a lot of skill set as well.